Hi Dougie, so good to meet you the other day. Here is the long-awaited soil sample. So yeah, this has been sitting in a bag, getting a bit anaerobic, so it's not a true depiction of what actually your land would look like in terms of the microbiology. But we'll give it a crack anyway. I'm happy to come up at some point in the future and actually get a more accurate picture of what's going on. Maybe I can actually bring the microscope to your farm or something like that next time. So this is from, it's not actually from the field right outside your house. It's from the one that's basically down by the Forth. So it's got like the reed beds running along it. So yeah, that's the one. So it's kind of like heavy clay, like I think much of your land is. So I'll just give you like a brief kind of 101, and we'll just do like a quick scan through the soil, see what we can find here. Camera's going out of focus for some reason. There we go. So yes, this is indeed your soil, and this was diluted about 1:5 dilution with just some water. The water has been treated with humic acid to make sure it's not chlorinated, to make sure it's not killing any of the microbes with chlorine. And this is at times 40, which basically gives you, sorry, hold on one second, something weird happening with the microscope. There we go. Yeah, this is 40 times what your eye can see. So it's zoomed in that much. At this sort of magnification, we're just like looking for filaments, strands of hyphae, basically. You'll see larger things like mites, arthropods. You'll see some species of nematodes as well, especially predatory nematodes. Predatory nematodes meaning that they feed on other nematodes, basically. So it would be very interesting to hear about the history of your farm, especially if you have any history of pests and diseases, any particular problems related to that, and see how this a functioning soil food web could actually give you some gains in that respect. So yeah, let's just have a wee zoom in, then we can see a few things moving. So I can immediately see that the soil is quite it's consolidated when you look at it with the naked eye, but it's kind of unconsolidated underneath the microscope, so all of its constituent parts are still broken up. So that does indicate low microbial activity, because usually these mineral particles are used to build, to pull organic matter together and used to create homes by the microbes effectively. And it's that movement of organic matter that creates structure within soil. So you can think of soil being exactly the same as like a coral reef in an ideal situation. So it's all biologically engineered structure. And it's the movement of particles and organic matter throughout the profile by biotic means rather than abiotic means that creates soil structure, aggregation, etc. So yeah, I'm seeing a lot of sort of mineral particles. The reason that they pull organic matter on top of these mineral particles and glue them all together is because the mineral particle is just as much a food source as the organic matter particle would be. So a lot of trace elements can be found within the crystalline matrix inside sand, silts and clays. And these are accessible, like I was saying before, by the enzymes produced by the microbes, so fungi and the bacteria. So I would say that on first sort of visual assessment of your soil, it's heavy clay, and to me it looks like it's collapsed. So that means I'm sure you know this, but like all the clay platelets are stacking on top of each other. So it'd be interesting to see what your calcium to magnesium ratio is, if you've ever had that done. I'm sure you have if you've had clay soil. But yeah, we probably want to boost that up to, aiming for a 7:1 kind of ratio. And then you can get flocculated clay particles where they stack, they sort of T-pose on top of each other and give more pore space, allow for gas exchange within the soil profile. So yeah, as you can see, I was talking about fungi and how key they are. It's not that they're more key necessarily than bacteria. It's just that they're the things that are often missing from agricultural systems, and we get huge plant health benefits and soil benefits from having from most production systems. We're looking at about a bacterial to fungal ratio about 1 to 1, and this in terms of biomass, in terms of weight, and this, yeah, at that point you're talking about efficient nutrient cycling. We of course want fungi because of their mycorrhizal properties, of course, as well. So they can tap into the roots of your plants, and they can be a symbiotic exchange of nutrients. The fungi can massively increase the root density of your plant. So I'm just having a look at this. This looks like it might be a fungal strand here. Let's have a wee look there. It is indeed a fungal strand. So that's what one looks like. Very short one, but that is a fungal strand. So you can see that it's kind of cracked off at 90-degree angles, and it's got like a cell wall there, what they call like a septa. So that's a division between each length of cell growth. And we can also see bacteria at this kind of scale. So this is times 400, basically 400 times what you see with your naked eye. And you can see it's teeming with bacteria, which doesn't surprise me. I would expect it to be a bacterially dominated soil. And there's nothing wrong with bacteria. Bacteria are great. Just also to give you another sense of scale, if you see one of those sort of black dots that's moving around, that's a bacteria. And one of the perfect circles is about 1 micrometer in diameter. So that's 1/1000th of a millimeter. That's to give you a sense of scale that we're looking at here. So very, 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 very small. Just above, just directly north of that fungal strand, we've got like a little diatom there. You can just about see it's the husk of a diatom. So a diatom that's died. It's made its outer shell out of silica. And generally speaking, they're photosynthesizers. So they're capable of moving throughout the soil profile and photosynthesizing as they go along. So yeah, all these bacteria are great, but I would imagine because the clay, the O horizon is so collapsed, I'd imagine a large percentage of these bacteria are probably facultative bacteria, which means that they have two sets of enzymes, one that works in low and one that works in high oxygen conditions. So they're going to outcompete basically aerobic bacteria, or obligate aerobic bacteria, that is. So yeah, it's interesting. The other thing that is very often missing on farm systems are nematodes. Most nematode species are highly beneficial to you. Predatory nematodes, for example, actually preferentially attack and eat root knot nematodes, and root feeding nematodes in general are predated by them in preference to other nematode species. We have fungal feeding nematodes as well, and we have bacterial feeding nematodes, and these are all really helping to cycle nutrients throughout the soil. From the bacteria or their prey item that they consume, they're going to excrete those nutrients in a plant available form, generally speaking around the rhizosphere of the plant's roots, which is where much of the action is happening. So just to give you like a sort of like a little prep on that, effectively, plants are obviously photosynthesizers, the primary producers in a system. They're responsible for sort of kick-starting the microbiology, generally speaking, within soil. And a lot of the material, a lot of the carbon that they get from photosynthesis, they actually excrete around their root systems out into the soil, and this is to attract species of microbes that they have evolved at symbiosis with. i.e. the microbes that they need to grow and function healthily in a natural system, so in a system that's not managed in any way by human activity, so without any synthetic inputs of any description. And these microbes, they are attracted to the plant's roots, generally speaking the bacteria first, and then any fungal spores that happen to be there will be stimulated to germinate, and they will grow towards the plant roots. If they're mycorrhizal fungi, if they're endo or ectomycorrhizal fungi, they'll form an internal or external network of hyphae around the roots of the plant, and then they'll grow in opposite direction. There'll be some sort of exchange site set up where an exchange of nutrients in exchange for sugars and amino acid from the plant will take place. So that's how the plant can get a lot of its nutrition. For the bacteria, they're attracted by all these root exudates. They're feeding off the root exudates, and the nutrients that they have in their bodies are generally speaking cycled by various predators that feed on bacteria. So it could be bacterial feeding nematodes, like I was mentioning earlier. A lot of it is done by protists, which are generally speaking single-celled organisms, like ciliates and flagellates and amoebae that you've probably seen before, and they predate these bacteria, and they excrete out all sorts of forms of plant-soluble, plant-available nutrients. The other quite interesting research that's being done at the moment is on the rhizophagy cycle. James White and all of his PhD students, so they've actually discovered that all plants are actually carnivorous. As far as they know, all plants are carnivorous, not just the carnivorous ones. So that is really entirely fascinating research that's being done. And sorry, the microscope stage just seems to be it's a bit broken actually. It's getting a bit too much harsh treatment recently. So yeah, the rhizophagy cycle, effectively and very simply, it means that bacteria and fungi enter into the plant roots, and they're held within the plant. The plant cycles them round with cyclosis, using its phloem and xylem, and effectively farms microbes within its body, microbes that it finds to be useful, to be symbiotic to it, to that plant species. It farms them to an extent where it can build up the population of fungi and bacteria and viruses and archaea within its body, and then it can release them out in its root tips and its root hairs back into the soil, back to farm minerals, essentially, go and collect trace elements from all the minerals in the soil profile and bring it back to the plant again in exchange for carbon. So yeah, very simplistic way to put it, but very, very interesting work. And the plant will actually consume and eat some of these bacteria as well that it has in its body. It'll actually zap them with superoxide and consume them internally. So that happens with wheat and with oilseed rape and most of your commodity crops. That's happening if the plant's given the chance anyway. So yeah, essentially I would say that this soil, we're basically looking for, if we're looking, generally speaking, where grain crops lie in an ideal ratio of fungal to bacterial biomass is about 1:1. So that would look like a single strand of hyphae at times 400, for example. So we're seeing very, very few hyphae. We're definitely deficient in that. There's also nothing really moving. There's no protists. They're very, very quick and agile. They are predators after all, so they kind of move like they're kind of effectively the lions of the soil. So we're not seeing enough of them. They're really key for nutrient cycling as well. And then this kind of idea of course, just like a human body, if a human has adequate nutrition, it doesn't have any deficiencies that are caused by excesses, because often plant deficiencies in terms of plant nutrition are caused by excesses of something else, which locks it all up. If a human has no deficiencies of what kind. They're not susceptible to disease. So the same goes for plants. So if we can make sure that nutrient cycling is happening efficiently and quickly, and in a natural way, then they can build up their own bodies to be resistant to diseases and pests, even aphids and grasshoppers. The other thing is, for example, if you have problems with, mildews or molds like ergots or something on your wheats, all plants, not just below ground but above ground, they also want to have a nice covering of biology, so a nice biofilm of beneficial biology that's going to act as like a physical barrier before any molds can actually reach the surface of the leaf or surface of the stem and start drilling in and causing damage. So they actually have if you have enough biology in the soil, as the plant germinates, it's going to coat its leaves with biology, and these can act as an initial barrier to pests and diseases, and we want to maintain that throughout their growing season. So quite interesting. I will maybe send you a link to maybe something that we want like our soil to look like underneath the microscope in terms of the biological community there that we would we'd be looking to build up over time. So let me know your thoughts. These are the beginnings of aggregates, by the way, these dark things here. So they're composed of some fulvic acids and some humic acids. I'm not seeing many ulmic acids. They're the kind of first chain of secondary metabolites produced by biological activity. But that is one. Let's just have a wee zoom in on that, see what that looks like under 400. So there you go. We're expecting to see more structures like this in a nice, nicely structured, textured soil. And these are held together by all the glomalins and glycomalicks that fungi and bacteria produce in order to make their homes. I think the end goal is basically to let nature do all the work for you. If you can set it up in such a way, and you can, aim to initially transition a single field. That, for example, looks like it's some sort of ulmic acid that's being produced by something. It does kind of look like a fungal structure there. I can't confirm that because it's a bit obscured, but I think that might be a fungal structure. Hard to tell, but potentially not a beneficial one. But yeah. The other interesting thing, I don't know what your pest pressure is, but in terms of fungi anyway, there are no obligate pathogens that we currently know of. So that means that given the correct environment, a fungal body can behave like a pathogen and cause damage to your crops. But in a different environment, it can behave as a mutualist, for example, and it can behave sympathetically and symbiotically with your crops. Quite interesting there. This, by the way, here is a testate amoebae, so the round thing, the thing that's kind of oblong here, that's a testate amoeba. You can just about make out at the left-hand side the entrance to its shell. So I don't know if you've seen amoebae before, naked amoebae, but they're kind of an amorphous shape. They can change and contort their body into any shape they want, and they're quite active hunters throughout the soil profile. That is essentially one of those, but stuffed inside a shell like a snail. So that's its shell that you're seeing there. You can just about see the sort of its body internally there. But they will be predating on spores and bacteria for sure. But it's hiding away in its shell at the moment because it's been disturbed. So that's quite interesting to see. Well, a lot of these protists, they eat something like 10,000 bacteria a day. So we need really, really quite high numbers to be able to sustain a population. What are you? That could, I don't think that is a fungal strand actually. And this thing here, This thing, I believe, is a fungal strand, but it is either dead or it's not terribly beneficial. It is indeed a fungal strand. It doesn't have any cross walls, so that's kind of a bad sign. It's very, very thin. That can be a sign that it's behaving like a pathogen. It's transparent, doesn't have a nice dark color. They get their dark color from melanin, by the way, just like we tan. They use that same compound to protect themselves from UV. Some of these more beneficial higher order fungi. So no, it doesn't look like that would be counted as particularly beneficial. Probably counted as no mycete, I would say. There's another diatom shell right in the center there, that perfectly symmetrical thing. But again, it's not living. But yeah, it's very common to find large. It's most likely you look at is a graveyard of diatoms. So that's fine. They're extremely recalcitrant bodies, so it takes them an incredibly long time to break down. So diatomaceous earth is just made of diatoms. It's an interesting structure here. This is a bit of plant material, I believe. We'll have to confirm that with somebody, but I'm pretty sure that's just a bit of plant materials, no cross walls. It's far too thick for most fungal strands. It looks like you can actually see the internal structure of the phloem and xylem in there, which is quite interesting. Not come across those particularly often. But yeah, you can see all the sand, silts, and clays. A lot of silica in your soil, a lot of sand in your soil, surprisingly. All these see-through transparent plates are silica. But you wouldn't necessarily know that by feeling the soil. This object here is actually, could be an actinobacteria. Yeah, I believe that's a strand of actinobacteria. They're facultative organisms as well, so able to cope with changing oxygen conditions. What I mean by oxygen conditions, it's like 4 parts per million to 6 parts per million. Those are the two highs and lows. So anything soil that's aerated, as it were, we don't really want soil to be aerated, but we don't want it to be as high oxygen as it is in the atmosphere. But something like 6 parts per million oxygen within the soil would count as aerobic soil, and anaerobic would be somewhere in the ballpark of 4 parts per million. So yeah, I will send you a video of so maybe fungally dominated compost. I just did a video for another guy, and you can just do a side-by-side comparison. This looks like, what's this? Another fungal strand, I believe. Could be an ex-nematode as well. I wouldn't count that if I was actually doing a bioassay. I wouldn't count that as a fungi, but it's interesting to speculate. But yeah, when I'm doing a bioassay, I'm interested in active organisms, so things that are actually alive and functioning, because that's what we're interested in when we're trying to do an analysis of soil. We're not interested in things that are dead or dormant. We're looking at things that are actively taking part. So yeah, I think moving forward, to use that horrible managerial term, if you guys are interested in doing anything like this, I would, or interested in, I don't know, different management in the field, just to see how it goes and trying to use some sort of biological approach to growing, then probably one of the easier things to get going, first establish if you got a plow pan. That's quite fundamental. You want to address that before you start planting any cover crops or any of these other sort of regenerative techniques. Definitely explore whether you can do a permanent cover crop, because that's really the holy grail. If you can manage a permanent cover crop and still grow your cash crops in amongst that, so you're never having any bare soil, you're never having any fallow fields, that's where you're going to get some real benefit. And obviously that's like, it's a difficult one to square off, basically. I don't think anybody's quite cracked cover crops yet, especially not using herbicides. But anyway, so yeah, if you've got a bit of time, I know you're super busy. Yeah, maybe just send me like a small list of maybe problems you're having, if you're having any problems. That'd be really interesting to know, like any lists of plant diseases, pests, anything that's bothering you that way. I'd be really curious to find out about that. Yeah, any sort of general issues that you've had over time. I know you're not necessarily keeping track of yields, but it'd be interesting to know, have yields increased or decreased over time, or yeah, that would be really interesting to know. If you, I don't know how far you can go back in the timeline of your farm, but yeah, I'd be really curious to know about that. So that's enough of my rambling. So good to meet you guys, and take care, man. All right, all the best.